Okay, so our last speaker for our panel is Colin Ophus, and while he is getting his screen shared, I will introduce him. Colin is a staff scientist here at Berkeley Lab at the National Center for Electron Microscopy, or NCEM, and he works as a research program as well as a personal user program focused on TEM and STEM and computational microscopy and material science. Has given many lectures and workshops on this topic, and we'll get to see one today. Just to highlight a couple of awards, he received the DOE Early Career Award in 2018 and the Burton Medal from the Microscopy Society of America in 2022. So with that, Colin, the floor is yours.
Thank you for the kind introduction. So very, very minor correction to that introductory paragraph. I am at NCEM right now, but not for very long. Yeah. I am switching over to the dark side. I've joined Stanford University. I'm out at the end of the month. So that being said, I still want to advertise for my former facility here in the molecular Foundry. If you guys see any experiments in my talk, anything that you're interested in, please do submit a user proposal, just like ALS. In fact, we even take partner proposals between ALS and the Foundry here. So our next cycle will be in September 2024, or sorry, their next cycle. Okay, so TEM is what I'm going to introduce here. The transmission electron microscopy geometry is very, very similar to any optical microscopy method. We have a source here of our electrons, an anode to pull them out of the tip to make them free electrons. We accelerate them to some significant fraction of the speed of light, form the illumination condition with condenser lenses, then use a very powerful objective lens to demagnify the source down to sub-angstrom dimensions often, and then projector lenses to magnify to up to about 1,000,000 times magnification, and then finally a detector. So this is what the beam path might look like if you stick a sample in the path of the beam. This is what the electron beam looks like on the sample, and in this configuration, you get an image at the detector that just looks like a magnified image of the sample. So plane wave into plane wave. But one difference between X-rays and electrons, of course, is how flexible our lenses are. All we have to do is change the current, and we can focus any of the different parts of our path here. So we can take a diffraction pattern, or we could focus on the sample. And this is what I'm going to primarily be talking about today, these so-called convergent beam electron diffraction patterns, or scanning electron nanobeam patterns, or to put it in the language of the previous speakers, the Stickson type studies or typography. So why do we actually do TEM? Unmatched spatial resolution. So even back when NCEM, the National Center for Electron Microscopy, was founded, we were able to get essentially unit cell resolution, sub 2 angstroms. And this is with the 3-story tall microscope that was installed next door, about 1,000,000 volts, right? So somewhere in the ballpark of 0.005 angstrom wavelength. And this is an image of silicon nitride. This is taken on one of our newer microscopes that's just recently been decommissioned in 1998, and the exact same sample on one of our TEM microscopes, transmission electrons aberration corrected microscopes. So if I zoom in, you can sort of see the different generations here. And you can see why aberration correction was so important to us. It basically only gave us about a factor of 3 to 4 increase in resolution, but the average bond length of most materials is somewhere in the 1 to 3 angstrom regime here. And so getting down to half of an angstrom allows us to see that each one of those blobs, which is a silicon column and a nitrogen column, are actually 2 separate dumbbell structures there. So you can see the hexagonal silicon nitride sample, it's Si3N4, and you can even see the bridge nitrogens, the two A sites linking the edges of the unit cells there. So how did we actually do this? Well, aberration correction, that's the big one. Electron lenses that are round must have spherical aberration. So if you want to improve on that, you just break the symmetry. You take like a north-south dipole, tip it on its side, and then do north-south, south-north, north-south, south-north, etc. in a hexapole, quadrupole, octupole configuration. Stack about 100 of these guys, and you can simulate negative spherical aberration and produce a nearly aberration-free image. That it were so easy with X-rays. We also primarily, these days, have improved by using better detectors, so direct electron detectors. No more scintillators that convert electrons into photons. We go directly from electrons into CMOS semiconductor type chips that convert directly into electrical charge, but also computation leverage. And that's mostly what I'm going to show today here. So this is a picture of what those 3 microscopes look like here. And the one on the far right, the TEM 1.0, is installed. And after the session here, if you want to see it, just follow me back to NCEM, and I'd be happy to show you that microscope in operation. our new spectra should be installed by the end of the year here too. So the next generation. Even that one on the far right there is still 15 years old. NCEM has very, very old hardware compared to most microscopy facilities. Okay, so primarily what I work on is scanning transmission electron microscopy. Because of its flexibility, we can operate in imaging mode, and as I already mentioned, use aberration correctors to make sub-angstrom probes, but we can also measure our sample in diffraction space here. And so this is often useful for thicker specimens, as you're going to see in a bunch of my experimental examples today. We also modify the illumination condition of our sample. For example, we could put in phase plates or amplitude plates. My group makes both of these things, and that's sort of how we adjust the illumination condition. But we can also do spectroscopy. So we can use, in this case, this is an alpha energy filter. There's a couple different ways to make alpha or gamma monochromation for the beam. And then in the far end, after passing to the sample, a magnetic dispersive prism can act as a spectrometer. It can essentially fan out the electrons based on how much energy they lose. And we can measure everything from what I zoomed in there, the sort of phonon spectra, all the way up to band gap measurements, surface plasmons, bulk plasmons, or even edges for ionization events there. And then finally, all of these methods can be combined with tilting the specimen to do three-dimensional measurements here. So this is why we call it a synchrotron in a microscope. It doesn't do everything the ALS does, but quite a bit of it, and all in a single machine. Very, very flexible. Okay, so phase contrast imaging is really what I'm going to talk about here today primarily. Just realized though, I think this slide's out of order here. Yeah, it is out of order. Don't worry, I'm just going to fix my talk live. You can totally tell I made these slides way ahead of time, right? Okay, so this is just a quick configuration. Okay, there we go. Just sort of showing this scanning diffraction operating mode here. So we used to use single pixel detectors, in this case measuring the scattered electrons. These days, as I mentioned, a direct electron detector can take a pixelated image of our diffraction pattern, and we can record this for every probe position. So advance the probe, record an image. Advance the probe, record an image. So this is why I coined the term 4D STEM, this sort of catch-all term here, because we have a two-dimensional grid of probe positions, and we get a two-dimensional image at each probe position. So very similar to the STIXM or ptychography beamlines up at ALS. So if we don't have overlapping probes, so we use a small numerical aperture, this is a toy model showing what type of diffraction we get. So because our probe is sort of a couple nanometers in diameter here, it sort of acts like a selected area diffraction pattern here, where instead of moving an aperture over the sample surface, we're electromagnetically scanning our beam and recording a fingerprint here. And this is, as you can see, showing the orientation and crystal structure, at least the projected two-dimensional version of it, of each crystal here. So we can do a similar thing for amorphous materials. If I randomly displace all the atoms on the right-hand side and then scan through the sample, you can see our Bragg diffraction pattern is replaced by these sort of translation symmetry free patterns, these sort of characteristic amorphous halos. It's the same thing. It's the structure factor of the material, but this time it has radial symmetry or annular symmetry there because of the lack of translational order here. So it's also a fingerprint for the local atomic arrangement. And I just wanted to quickly mention too, if anybody wants to know how to make movies like this directly in Python, I wrote a quick tutorial here that's available in Colab, Google Colab here. And I even figured out how to render the movies directly in the browser window. So this is recorded, by the way, and I'm happy to give these links afterwards. So here's another example of the type of thing we could do. This is something I'm actually working on for a meeting tomorrow. We're taking a functional ferroelectric strip there, that's the colorful strip, between two titanium nitride electrodes on a silicon wafer substrate there. And we're trying to figure out which of these grains is the ferroelectric phase, and if they are the ferroelectric phase, what direction it's pointing here. So these sort of scanning electron nanodiffraction patterns or nanobeam electron diffraction experiments, very, very big business for us. We do a lot of that. I also wanted to show soft matter too. Here's two polymer samples where I reconstructed the morphology. And this is just done by, if you look at the raw diffraction patterns, inferring where the pi-pi stacking of this polymer chain material is sitting. And then you can see the difference between this P1 molecule without a chemical additive with the chemical additive here. So it completely breaks up the grain structure and adds a lot of overlapping grains here. Okay, so I'm not really going to talk about nanobeam at all for the rest of this talk here. I did just want to mention, though, all these different analyses are performed in my group's Pi40 STEM analysis toolkit here. And we've recently started adding X-ray modules to it. So we've been doing X-ray ptychography recently with this code. And it has a whole bunch of different analyses. Here's our core developer team. Everybody is in town right now except for Steve and Alex, I think. Ben is back for the workshop tomorrow. And Steph and George are postdocs at my group there. So it can measure all sorts of different things from 40 STEM patterns. We got an R&D 100 award last year. And I put a whole bunch of our recent tutorials from our conference a couple of weeks ago on Google Colab as well. So if anybody's interested, they can check them out there. Okay, so now is where the phase contrast imaging slide should be. So I was specifically tasked in this workshop for talking about why coherence is important and what it enables. Well, in a nutshell, phase contrast imaging. So this is kind of an old result, plane wave HRTEM. And what you can see here is a grain boundary between two sheets of single layer graphene. This example really shows why we do phase contrast imaging. It's extremely dose efficient. And one great thing about electrons is they interact incredibly strongly with the sample. One terrible thing about electrons is they interact incredibly strongly with the sample. It's kind of like sandblasting a soup cracker. Our electrons will obliterate things that we're imaging at atomic resolution. And that's why we need phase contrast imaging methods, because they can be up to about 50% efficient. Whereas the incoherent imaging modes or nanobeam diffraction imaging modes, they're usually ballpark 1 to 5% sort of diffraction efficiency or imaging efficiency. So in STEM, the easiest way to do our measurement here is simply to make a probe that's down subatomic scale. And then when you go near an atom, the atom, which is positive electrostatic potential, will deflect the negatively charged electrons towards it. So this is why we call this differential phase contrast, because you get a different signal left versus right. And you can also pick up electric fields, extrinsic in this case, or magnetic fields. And these two are how we built our microscopes on the right. And I'm not really going to talk about them further here. So what does it actually look like? Well, if you scan your airy disk function over a step or a larger step there, you can see the response in diffraction space in the far field underneath the sample. So all we need to do is position either a segmented or a pixelated detector, and we can measure that shift. So let me show you an example here with a small numerical aperture. If I were to scan through an atomic resolution nanoparticle sample here, you can see when I enter the nanoparticle, I get diffraction to the Bragg vectors there. But even though the phase is changing as I scan through the atomic column, that's the color map shown on the bottom image. The middle panel shows that there is no change in the diffraction intensity. So the left-hand side diffraction, the right-hand side diffraction, equal intensity no matter where the beam is positioned. So if we measure that green line, the position result center of mass shift of the probe, we measure, essentially it's proportional to the gradient of the potential. So all we have to do is take the FFT, use anti-differentiation or integration here in Fourier space, and we can reconstruct in red the phase. But we want to see atoms. So what we can do is boost the or open up the numerical aperture, repeat this experiment, and now every time I enter the sample here and I go on both sides of the atomic columns there, you can see we're going to alternate between constructive and destructive interference and pick up a little bit of a shift there. So now if we integrate it, we're recording some of the atomic potential there. If I really open up the numerical aperture, so forgetting about aberrations for now, this super, super sharp probe is exquisitely sensitive to the atomic potential now. And so now I can reconstruct the entire sample potential, so the low spatial frequencies and the high spatial frequencies here. So why don't we do every experiment with a massive numerical aperture? Well, in a word, signal to noise. If I only put 500 electrons into my probe, you can barely see those dots there. You can see we get these strong artifacts in our reconstruction. And as I shrink the numerical aperture, the artifacts are reduced as well. So we have to balance real space resolution against signal to noise and precision here. This is essentially a Heisenberg uncertainty relationship here between the real space and Fourier space here. Okay, so what does it look like for a real sample? Well, for every probe position here, we measure the deflection. This is the shift in X, the shift in Y. We have to do some alignment here, which I'm going to skip over right now, but we figure out what the precise reciprocal space rotation versus our STEM scanning direction is and correct for it, and then we can reconstruct the sample. And so all we have to do is this Fourier integration that I mentioned, and voila, we have our atomic potential here. So this is a twisted bilayer sample. I actually dropped the citation. This is from David Muller's paper on ptychography, I believe in 2018, published in Nature. I'll fix this slide after. So here's another example. This is from Pete Nellis' group over in Oxford. This is what DPC looks like for a carbon nanotube with these buckyballs and iodine atoms inside. And this is just a different form of non-iterative ptychography to compare. So we do iterative DPC, center of mass, because we want to solve the boundary conditions of our sample. Otherwise, it basically is reconstructed in a single iteration here. And so I'm not going to go into the details of a complex DPC study here. I'm just going to highlight two panels. Panel E is the gradient of the electrostatic potential, meaning this is the electric field. And panel F is the same thing calculated by density functional theory. So this is just to show that in a very quantitative way here, we can measure electric fields inside our sample. Now, I do want to emphasize, we only very weakly pick up bonding. Most of the electrostatic potential is the ionic core. This is another big difference between electrons and X-rays, right? We're essentially scattering off of the screened ionic core. So we mostly probe core electrons. Valence electrons are a really tiny perturbation off of those core electrons. And that's why you can sort of see a couple systematic errors there at the interface. But overall, it's not bad. It works pretty well. Okay, so what if we defocus our probe here? So if I scan through, I don't know why it's so, I think Zoom is just really laggy. It's much smoother on my screen here. So this is showing if we move the crossover out of the plane of the sample, essentially a shadow image of our probe convolved with the phase curvature of the probe, the aberration surface of it, right? And so what you can notice is that it's essentially an image of our sample multiplied by a contrast transfer function. So if I were to look at another synthetic example here, a 3D plane wave here, and I were to take a single pixel in my reciprocal space 40-STEM data set here, and I make a virtual image from it, and then I move it around pixel to pixel here, you can see that each one of these sort of pixels in our cone of illumination angles is intersecting the sample at different angles here. So it's picking up three-dimensional information, essentially. And this, if you've done Fourier ptychography, this should definitely make you think of Fourier ptychography here. And so what we can do is do a little bit of alignment, parallax transform, apply a Frañó transform on top of that, and essentially depth section our sample. So this is just what we call the parallax module in Pi40-STEM, and it can recover 3D information and in focus images of our sample. So I do want to quickly figure out how to stop sharing. And just quickly make a quick plug. I founded a new journal last week called Elemental Microscopy based on open source interactive Python and Jupyter notebooks here. And we wrote an article here called Tilt Corrected Bright Field STEM about this parallax imaging. And I specifically wanted to quickly pull it up here. Let's see if I can do this, just to show why we founded this journal. Specifically, if you're trying to explain mathematical concepts like this, right? We have Python notebooks that do the reconstruction. But when we align all of these virtual bright field images, you can see we can bring our sample into alignment and reconstruct. This is a biomolecule in this case, and we can actually see the effects of what if we were to defocus it, or what if we were to apply astigmatism, what if we were to change the angle of the astigmatism. So these sort of interactive things is a lot of what my group is working on right now. And a lot of our talks going forward, we're going to eschew PowerPoint altogether and switch to giving talks like this. So more on this if people are curious, feel free to ask me questions after. Okay, so returning back to our actual sample, simulations are all well and good, but come on, PowerPoint, you can do it. Spinning beach ball. Oh, that's what I get for doing a live demo while Zoom is running. There we go. So this is just comparing when we have a defocused probe here. This is the incoherent dark field image versus the differential phase contrast image versus doing this parallax imaging here. So similarly, if we spiral out, we can see all of those plane waves hitting the sample at different angles. We align it and produce an aligned bright field image, fix the contrast transfer function so we get positive electrostatic potential. But very importantly here, the resolution is limited by the numerical aperture of the measurement, right? Not by the real space sampling. So if you only limit yourself to the real space sampling, you're cutting off information in Fourier space. But if we upsample it, we can recover those Bragg diffraction peaks and get some atomic contrast back. Okay, so I'm not going to talk about the iterative ptychography method here. Essentially, we just use the forward operator, do a difference map, and then back project it using the same equation here, the adjoint operation. So we have an initial guess for our probe, a position of all of our scan points here. We measure the simulated object multiplied by probe, compare that to experiment, take the difference, back project, iterate, 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 and you get atomic resolution reconstructions here. So this is going back to that same data set just to compare the resolution here. You significantly improve an aberrated probe reconstruction by going from DPC to parallax. Again, you can think of this as Fourier ptychography, but iterative gradient descent ptychography absolutely annihilates it here. So this is why we use gradient descent ptychography for virtually everything where resolution matters. And I have this slide comparing the different methods. No time to walk you through this insane figure right now. Feel free to look it up on the archive if you're curious. Okay, so this is just looking at some biomolecules here. This is one of the reasons we're pursuing these methods is to try and see, can we do biological protein reconstructions using them? And the answer is we can. So by combining with cryospark and plywood stem here, I think we currently have the resolution record for stem reconstructions of proteins. This is primarily done by Henning Stolberg's group over at EPFL. My group just provided all the software for the back end here, other than the bio-cryospark program here. But it's not that impressive. We're at about 5.8 angstroms resolution, whereas TEM is down to 1 to 2 angstrom resolution. But considering we've only been working on this for about 6 months, I'm not too displeased with how we're doing so far. Okay, so 2 other quick examples. Again, just comparing conventional STEM to ptychography. This is boron nitride, a couple sheets. You can see the single atom defects very clearly in the ptychographic reconstruction, impossible in the incoherent conventional STEM image. And I think one of the early speakers mentioned multi-slice ptychography. We have our own implementation of it. It works really well. This is a twisted bilayer of strontium titanate, and we can easily measure the top versus the bottom or sort of interfacial slices here. This technique has a resolution somewhere in the ballpark of 1 to 4 nanometers along the beam direction. So not atomic, but good enough to do specimens like this. Okay, so 2 more quick topics for the rest of my talk here. How do you actually recover 3D information? Well, I alluded to it earlier. We do tomography. So this is how we used to do it with incoherent ADF STEM imaging ptychography. We simply tilt the particle, record an image, tilt the particle, record an image, and then use computational methods to reconstruct the 3D structure. And along with John Miao's group at UCLA, we published a bunch of these reconstructions at atomic resolution over the last 10 years here. So this is a pretty mature technique right now, but I already mentioned it's extremely dose inefficient. So one thing my group really wanted to do was replace the ADF image with ptychography, and we had done it. So here is a Alex Settle UC Berkeley sample. This is an encapsulated double wall carbon nanotube with zirconium tellurium nanowire inside. The structure, super complicated. If you look at any 2-dimensional projection, you can't figure out what it is. So we set out to solve it. And again, just comparing the basic methods here, ADF incoherent imaging, differential phase contrast, and ptychography. This is why we picked ptychography. It's quite challenging. Philip did a lot of tilts, a lot of ptychographic reconstructions, but he was able to get images of the tube along many different directions. And here's a class average image of the 3D volume. And you can see that we've reached true 3-dimensional atomic resolution. So I worked with Sinead Griffin here in the Foundry to confirm the model. It's quite bizarre, the atomic structure that we solved inside. So first, the outer shell structure. These are zirconium tellurium 5 structural units. But in regular zirconium tellurium 5, the bulk phase, each of these pentagonal units would be up, down, forming a 2-dimensional sheet. Here, they're aligned in the same direction, forming curvature on the outside edge of the tube, and the tube is highly elliptical. So it's incredibly unexpected. We definitely expected a circular nanotube here. The core is even more strange. This zirconium tellurium 2 phase has never before been observed, but we did confirm it's stable under DFT at least. And actually, if you take this ZRTE5, you take the 2 tellurium atoms that are furthest away from the core, rip them off, that's the zirconium TE2 structure. So it does seem logical. And we found individual chains of tellurium. Tellurium is one of the only atomic species that are known to be able to form 1-dimensional atomic chains. So it actually seems quite reasonable. I don't think this is the final word on this structure, but we've demonstrated that ptychotomography can be used to solve structures here. Of course, the real next step, joint reconstruction. And so we have a couple different code implementations of this full three-dimensional joint ptychotomo. But I need some help scaling this up to multiple GPUs if anybody is interested. We have a tutorial online. It works pretty well, and it can fill in a significant amount of the missing wedge because it's just much more efficient. It's using the 3D information of multi-slice inverse or inverse multi-slice typography. Okay, one last topic. How long do I have?
2 minutes.
Okay.
3 minutes.
So I was asked specifically by the organizers to talk about our adaptive optics, in this case, a phase plate. So this is a cross-section showing scattering angle from 0 out to about 2 angstroms or 1/2 inverse angstrom. And you can see this is what we would normally sort of see in our imaging conditions where we're using a negative defocus to balance a positive spherical aberration. So the goal is to make the CTF flat, right? The contrast transfer function here. So we can open up the numerical aperture and do atomic resolution imaging. But we're limited because that spherical aberration, C3, shoots off to positive infinity with Q to the 4 here. So what can we do? Well, what we could do is cut out a little strip of this and then apply a linear correction from a positive to a negative terminal there and correct the electrostatic phase shift of that spherical aberration. And we can do this in many, many different segments, flattening the phase, keeping it between -π/4 and +π/4. So what we have tried to do is design a corrector here that has this sort of annular circular symmetry and then can apply from 2 terminal phase shifts here a correction to the wavefront. So negative spherical aberration. We did a whole bunch of calculations to sort of optimize the geometry of these things. And we worked with the Peter Dennis group to actually fabricate them. So some quick simulations just to show. If you have a small numerical aperture limited by spherical aberration, no atoms, put in one of our you see atoms, or correctors, excuse me. It's not nearly as good as the ideal perfect wave, though I should point out. Like these faceplates still have some drawbacks here. In particular, we can't apodize our probe, meaning we don't have order sorters along the beam direction. We only have 3 condenser systems. We don't have another crossover that I could cut off the other orders here. So we've actually made these things. We're in the final step of installing them in a microscope. I don't think they're going to work as well as our current correctors, but our current aberration correctors cost about $1 million. And these are going to be like $10 a chip or something like that. So this is part of my ongoing goal to try and democratize atomic resolution imaging here. Okay, so with that, I will wrap it up and just say, if you're interested in these techniques, I wrote a review paper about 5 years ago now. All of our code is online on GitHub, along with many tutorials, hundreds of test data sets. We're all on Slack. We're always available to chat. And we wrote a paper on our phase contrast methods in our code, which is currently on the archive and currently fighting with referees to get this accepted. Who would have thought that a method paper would be contentious? And with that, I will thank my many, many collaborators and be happy to take questions.
Right. Okay, questions?
Thank you. Cryo what? Same as cryo what? It's meant to be used for cryo-EM, yes. So the plane wave imaging that most biologists use, there's 2 big software packages, RelyOn and CryoSpark. So both of those packages take class images of proteins, merge them in 3 dimensions, and solve the protein structure. So our code got as far as making the class images of the proteins, which we then passed into CryoSpark to solve the structure. And I say we, I really mean Burke, the student from EPFL, who did all of that work. There's a couple new packages. In particular, I think Chan Zuckerberg, UAU, is working on some of them here. Packages like CryoDragon that use machine learning. We also, by the way, I have a ton of machine learning versions of all of these things too, which I didn't have time to show today. But yeah.
Awesome. All right, any other last questions?
I was curious, given the flexibility of your lenses, right? Although with the variations, with limitation and so, can you define a willing and known way and then apply recover images? Essentially, can you perturb voluntarily can you create a controlled, if you prefer, aberration and then use this to sort out, for example, for let's say reconstruction problems. So
Yeah.
to reduce symmetry problems, right?
The answer is yes. So we can't modify most of the column because it's purchased from vendors, unfortunately. But with an adaptive faceplate, we will have the power to do that, to do wave function tuning. And you've hit the nail on the head, right? If you put in phase diversity, it improves ptychography. And you can go a step further. You can adaptively solve the wave, or you can make orthogonal imaging conditions or symmetry-based conditions to pick up really, really subtle features. We're hoping to do all of that if and when we can get this installed in a TEM and get it working. We're so close. We have the chips. We just have to finish the aperture rod and electronics installation. I wish we had 10% of the engineering knowledge of ALS. Hopefully that answers your question.
All right. Okay, let's thank Colin and all our speakers once again.